Okay. Hello, everyone. Thank you for coming to IFAS AI and Data Science Seminar Series. Today, we have two speakers, Dr. Henry Medeiros and Dr. Arnold Schumann. Our first speaker is Dr. Henry Medeiros. I'd like to introduce about him a little bit. Dr. Medeiros is an associate professor in agricultural and biological engineering at the University of Florida. He received his PhD degree from School of Electrical and Computer Engineering at Purdue as a Fulbright scholar. His research focuses on development of AI models and robotic systems to improve agricultural production. He is an associate editor of Elsevier's Smart Agricultural Technology and senior member of IEEE and recipient of National Science Foundation Career Award. He has co-authored more than 50 peer-reviewed publications and holds several U.S. and international patents. Now, floor is yours, Henry.
For the introduction, Anna, it's a pleasure to be here today. So I'm going to talk about generative models, right? So today we have a generative models session. So this, I'm going to present first, just some kind of preliminary results. So just to make sure you can see my screen, right? I'm sharing the right thing.
Yes, we can see.
Okay, thank you. So this is just some, initial results that we got on this collaboration that I recently started with Vivek Sharma, right? So we're looking at, applying generative artificial intelligence models to agricultural problems in general, right? So this is kind of some preliminary results on this heat stress estimation problem model, I guess, using aerial images that, again, Vivek and I started working on recently. So if I give you the impression that, I have any, deep knowledge of irrigation, that's not true. This is all Vivek's. And also, I can't claim any merit on any of the results you see here either. They were all generated by Nesrin Ben Hassin. She's a postdoc with my team. So the only reason I'm the one speaking here today is because this is very, very preliminary, right? And I wanted to share this with everyone and see what, if you have any thoughts or, ideas on how we can kind of build upon this. But of course, I didn't want Nesrin to have to come here and potentially have to defend her or our, preliminary results. So next time you'll be hearing from her directly. So, so we're trying to come up with a solution to this dynamic variable rate irrigation problem. And so the idea here is that, let's say you have we have variable rate irrigation, right? One of the limitations with the current approaches is the fact that basically they are static, right? You generate your variable rate irrigation map at some point, and then you just stick with it, even though conditions change a lot throughout the season. So what we want to do is kind of come up with a dynamic model, right? A model that can basically observe the environment, adjust the prescription map, and then send back the commands to the system. So basically, we conceive this is kind of a high-level overview of what we have in mind at this point. I thought of this system that has basically a thermal image estimation model. We have a irrigation prediction model, and then we have the dynamic irrigation control component itself. So the idea is that the thermal image estimation model would collect, RGB images and thermal images. So we collect those image pairs across the season. We would also get some environmental parameters that are relevant. And then from there, we get that information and we train a model a thermal image generation model, a model that basically takes RGB images and spits out synthetic thermal images. But now, because part of what we would like to remove the need for collecting thermal images, and we'd also like to be able to not use a drone at all, right? We would like to be able to monitor the field more frequently. So the idea is to use cameras mounted in the center pivot to basically reconstruct a view that is similar to the UAV view. It's not going to be identical, but it's hopefully going to be similar enough that we can do like a domain adaptation for these models. But then this RGB image would go through the thermal image generator. And in addition to that, we would have like a plot segmentation model that would basically identify, the different plots and separate plot from field, so that we can kind of get this synthetic thermal image and the segmented crop plots, and then do kind of a thermal or a heat stress estimation per plot, right, individually. So the idea then is to send this information to this dynamic prescription map generator, which also takes, environmental information, just like the other models, and then uses those to create the prescription map itself, right? What's going to be given to the to the control system, right, for the for irrigation, and then we can send. Sorry, it's the question. Oh, no. So then once we send this control signals back to the field, we can do the irrigation, and while we do that, we can collect data that we can then use to kind of continuously learn, right? And we can use like self-supervision strategies or maybe reinforcement learning to keep the system self-updating. Of course, this is an ambitious goal. In the few months we've been working on that, we haven't done all of that. So in this, today I want to focus on the thermal image generation model, which is the first part of our proposed system, which again, it's meant to get these pairs of RGB and thermal images that were collected by an UAV. And one thing to keep in mind is that there's no, other than flying the UAV and collecting the data, there's really no manual annotation needed here, right? So those data, they are, how can I say, intrinsically aligned, right, by the data collection procedure itself. Now, those images, they're extremely high resolution, of course, right? They're like orthomosaics covering relatively large areas at high resolution. So training a model that can learn those huge images that contain very fine details, right? The problem is not just that the image is huge or that the details are fine. The problem is that they're both there, right? So we can't have having a model that will learn both would be challenging, and you would definitely need a model with very, very high capacity and a lot of data. So instead of doing that, we decided to try and keep things simple and train a model that learns to reconstruct image patches, right? So what we do is we get those very high resolution images, and by very high, I mean 19 by 10K or something like that. And we through a sliding window, and we extract patches from both the thermal image and the RGB image. And then those patches we use them in this generative adversarial network. It's a model. It's kind of one of the models that popularized this image-to-image translation ideas. It's called Pix-to-Pix. Again, an easy-to-use model. There are much more sophisticated alternatives today, but again, it's the idea is to try and explore this concept. So like any gun, Pix-to-Pix is going to have this generator. So we're going to train the generator to reconstruct those thermal patches, and then we're going to get those synthetic patches that the generator learned. And again, this is nothing special, right? It's part of how generative adversarial learning works. But the way we are using it is, so we're creating those synthetic patches. And then we have this discriminator whose job is to learn whether those patches are synthetic or if they are real, right? So, but the giving the discriminator just the synthetic patches makes it easy for it to learn, how can I say, I guess, synthetic artifacts, image generation artifacts that, they're just like problems with the generator that you can kind of memorize and just use those to avoid learning, you know, or to tell what's real and what's not, right? So what we do then is we make the life of the discriminator harder by giving it also not only the synthetic thermal patches, but we also align, you know, basically, you know, concatenate in the channels, the real RGB patches, right? So then the discriminator takes, you know, this, the training samples for the discriminator are the synthetic patches with real, synthetic thermal patches with real RGB patches, and the real thermal patches with real RGB patches. So, and the discriminator tries to tell them apart, and then, you know, by this adversarial learning, both the discriminator and the generator get better. And then in the end, after we get the all the reconstructed patches from our generator, you know, at inference time, we simply, you know, just kind of rearrange them and reconstruct the entire synthetic thermal image. So again, just to further emphasize, so we're not generating the entire thermal image. We have actually a model that generates image patches, and it's important to keep that in mind because you see some of the, I guess, consequences of going that choice and what we can do about it. So very briefly, you know, our data sets that we're using here. So again, they're part of the VEX data sets. So he collected this using a MicaSense Ultim camera. It has a resolution of 1.31 centimeters per pixel. So all the data that is available, we only use the dates when we had the pairs, right? The paired orthomosaic and the thermal image. So there are two different crops. One is corn, the other is potato. For the corn data set, we have 8 orthomosaics with the corresponding maps. And then for the potato data set, we have 11. Actually, strictly speaking, we have, I believe, 10 and 14 or 11 and 14, yeah. But we're saving a few images just for, you know, as our, it's called test set, right? So for now, we want to keep a few images apart. We haven't touched them yet, just because as we are happy with our results in our validation data sets and our model parameters, then we're probably going to go grab those images and further validate the model. Of course, hopefully by that time, we'll be collecting some additional data. So the corn data set's about 2.5 acres, the area that it covers. Data was acquired between August 2022. In the potato data set, the area is a little higher, as you can see. It seems like we're kind of, but the resolution, the sensor is the same, right? And then that acquisition here is from May to July. So again, our data sets are very small, right? We have 8 images only in the corn data set, 11 images in the potato data set. There's no way we would be able to train any adversarial model, any generative model, any model with just this amount of data, right? So that's part of how the, you know, this kind of data partitioning, you know, that the sliding window approach also helps us. Basically, if we are training a little patch reconstructor, then we have a lot of patches in these images, right? And so in our experiments, basically we use k-fold cross-validation. So for basically, you know, we train the model using, let's say in the corn data set, we use 8 folds, which is basically leave one out, right? So we test the model, or we train the model using seven images and evaluate it in one of the images. And then we repeat that 8 times, right, for all the 8 images. For the potato field, we used 5 folds. So again, it's like, you know, you partition those out of those 11 images, you know, you partition them so that you have, you know, 5 separate sets. And then, so we have 2 types of evaluation, right? So first, we look at the performance of our model with in the crop that it was trained at, right? So let's say we get the corn images, we train our model, and then we test it on the validation images of the of corn validation images. And then same thing for potato, train on potato, test on potato. And then we do the like a cross crop evaluation where we train on corn, test on potato, train on potato, test on corn. And we've been evaluating, it's part of this, that what's interesting about starting this new collaborations is that everything is an opportunity, right? We've been looking into several performance metrics that could reflect, the results we're getting. Today, I'm going to show only the root mean square error because it's easy to understand. And just to make it clear, here I'm talking about the root mean square error, where this y hat is a thermal a pixel in the thermal image that was generated by our model. Yi is an actual thermal pixel that was provided as the input for the model, and m is the number of pixels, right? So for each image, we have this RMSE value, and then the results that I'm going to report are the means of those image RMSEs and their variances as well. There's a lot of room for, looking into more granular performance metrics. We've been looking at things like, kind of patch-based metrics that have been coming up recently with that very purpose, right? With the idea that you want to be able to measure performance in an image considering the image structures, right? So one of the most important things in an image for us is that it retains its original structure, right? If you think about those plots, right, in the images that I was showing you, that's the general structure, right? So any performance metric should favor an image that has that overall structure. I'm not going to talk about that today because there's a lot more to be investigated there. But the RMSE results, they give us a pretty good indication of how things are working. And I might have mentioned that, well, the models, and this is in our kind of preliminary evaluation, we're using patches of 256 by 256 pixels. Arbitrary decision. So we're very likely to get better results with higher resolution patches and a few other tweaks that we may talk about in the Q&A. So this is just to give you a kind of an impression of what happens in the kind of single crop performance, right? So we have that input RGB image, and then on the left, right, and then on the right, we have the actual thermal image. So those were the images that were, those were not used to train this specific model because they were the validation set, but you get the idea, right? These are the images that are used to train. And here you can see the estimated thermal image. So you can see that it kind of retains the overall structure and to a decent amount. We see some interesting things right away, which is first, like there's some blocking artifacts that are caused by our the patching, the sliding window. We have a few strategies, like there's some kind of well-known strategies to deal with that. We've used that in some other problems, like, having partially overlapping windows tends to be enough to kind of, smoothen this, bringing the average window value among neighbors closer, and then it smooths that out quite significantly. Another issue that we're still kind of looking into is that, there seems we might need some kind of calibration, or to get some data points so that we can tell the model. You can see that the actual thermal image kind of has, how can I say? It's well, I guess maybe looking at the individual patches is a better way to discuss that. So you can see that here we have two rows, just showing some zoomed in versions of a few images. So as I said, the images are very high resolution, right? So you can see that our model kind of seems to, underestimate plant temperature, right? You can see in the green areas, it's a little darker than the response of the real model. And sometimes it kind of seems to overestimate the ground temperature, right? You can see that the model is a little. So I guess I'm trying to say the models need to be calibrated to the real data, right? Using real temperature sensors. That's part of what we need to do in the future. I'm sure that's going to help a lot. So here is a result of the cross-crop test. Right, so here again, our input is an RGB image of the potato crop. Here I'm only showing you the actual thermal image just so for reference, so you know what the estimated image should look like, because the network never saw any of that, right? So the network was only trained on the corn images. So this is promising. I guess obviously you can see that the quality of the reconstruction is lower. In particular, the blocking artifacts are a lot more prominent. Again, it's something that there are approaches to fix, but this is promising because it shows us that there's a lot of common information that can be learned across crops, right? So just to have some, try and quantify a little bit what we're getting here. So our root mean squared errors are on the order of 1 to the -3. So you can see here, in this box plot. So we have the model trained on one crop and tested on the other. So C versus C is basically trained on corn, tested on corn. C versus P is trained on corn, tested on potato, and so on and so forth. And basically, there are interesting things that we've learned so far, right, based on these experiments, but there's still a lot more to look into, right? So I guess one thing that we saw is that even though the appearance of the image is the performance is visible in the qualitative experiments, quantitatively the performance drops, but the errors remain low, right? So if you look at the table at the bottom, you can see that whereas the model trained on corn and tested on corn had an RMS of 15 to the -3. If we test that model on potato, we get just like 0.5 e to the -3 RMSE increase. So that's why, as I said, the metrics themselves are important. The structure preservation is something that we're aiming for in future models. And there's a lot of guns that try to address exactly those problems, right? So we have, again, this is just a feasibility study, I guess. So again, the qualitative analysis also showed us that not only the metrics, the structure preservation is needs to be looked into, but also the performance seems to be different between the ground and the crop. So it looks like we get slightly more stable results on the ground, which wouldn't be entirely surprising. But again, further topics for investigation. So just to wrap up, and so we have some time to discuss this. Basically, I tried to show you just one example of how we can use adversarial networks to learn to reconstruct thermal data from RGB data. This model, again, it's very simplistic. It's just an initial model, but I didn't mention that, but it is a conditional model, right? So you can have generative models that basically don't look at your input at all. Now, when you have your input parameters condition the model output, and then that's a conditional model. Our model is conditioned on the RGB image, but it could be conditioned on lots of other things, right? So we can add additional relevant information to the model, I would say, relatively easily. So of course, clearly we still need further experimentation, right? We're still trying to understand what are the operating conditions of the model, right? When is it going to work, right? How does it vary, as you change your training data over time, and yeah, of course, the factors that impact its performance. So even though we had a lot of image patches, right, we need to acknowledge the fact that we only had 8 corn images and 11 potato images, right? So it is a small sample, even if the patches are sufficient to train the models. But still, I think those results during encouraging. And one thing that I find particularly encouraging is this potential for like cross-crop pre-training, right? To allow like incremental model learning, right? We saw that training the model in one crop and testing it in the other is certainly not a waste of time, right? So there's a good chance then that, you know, we could get those model, like we could start looking into strategies to pre-train our models using, well, all the data we have available, and then fine-tuning it in, you know, specific scenarios, crops or applications, fields, whatnot, as needed. So I guess that's all I had for today. Be happy to answer any questions.
Thank you, Henry. Is there any questions? Please raise your hand if you have any question.
Thank you
yeah.
for the interesting talk, Henry. I have 2 questions. So this is Haipeng from Animal Science. So I think my first question is, you mentioned you are using the patches. So it's kind of my understanding, it's like a kernel with a square you loop through your images. Are there any overlap between when you move from first kernel to second kernel?
That is a very good question, Haipeng, because it has huge implications. Right? Yeah. So I guess the short answer, just to be direct, is the short answer is no, it doesn't. And that's a big reason behind those blocking artifacts that you saw in those slides, right? Okay. We have used that before in several other works where we were dealing with these very high resolution images. Like basically, you know, if you do a little bit of overlap, just like 10-15% of overlap among neighboring blocks, you get a pretty significant reduction in this blocking artifact. I'll be happy to share with you some of it. Yeah. Okay. The links. I'm saying that because that's exactly what we're going to do next, right?
Okay. Yeah, I think if you overlap, you can get more bad patches and more information. It may help improve your like performance. And yeah, my second question is because I can see your generated like thermal images. It's actually the temperature is a little bit different from the real one. But my question is, are you more interested to predict like generate image to estimate the real temperature, or you care about the differences? Your generated like images, you may have different the contrast differences. So I'm just wondering, are you care about the real temperature or like a contrast, the ranking within your generative images?
So I guess the short answer is I don't know. Then the longer, more interesting answer is I think we are going to need calibrated models, right? So I think we're going to need models that whatever image they generate can, you know, the pixels can be mapped to actual temperatures so that we can use this, you know, physical real world information in the other models. Because if you remember, the hope is that this model will then be used in conjunction with this plot segmentation model to train the prescription, the irrigation prescription map prediction model, right? So our hope is that we're going to calibrate this model. And what I have in mind, you know, is again based on some stuff we've done before, is to try and have a few temperature samples in the ground. Hopefully, we might be able to get away with using, I don't know, some weather station data, but I don't know. But if we could have a few samples in the ground, I think we should be able to calibrate the whole map. Okay, as part of the learning process. Does that answer your question?
Yeah.
But you touched on some very, very important points. We should probably, you know, get some coffee and discuss all the things I didn't say about your comments right now.
Okay, sure. Thank you, Henry. Thank you.
Okay, so due to the time constraint, we're going to have one more question. But Jose, if we'll ask Rafa first, and then Jose, if you have question, please just type it in the chat so Henry can respond to you later. Rafa?
Yeah, a very simple question. So the clear difference between the potatoes and the corn even the cross model does better than the potatoes alone, you know, so the corn and potatoes does better than the potatoes alone. So is that something related to the specific, the canopy, or the something that is structurally different on both?
So I guess we're hypothesizing, I guess, that it's just a matter of bad luck. You know, I think we have this model that we've been kind of, you know, tweaking to get it to work. We started with corn. So this is interesting because it addresses this kind of, you know, whole idea of community overfeeding of data sets, right? So we get, so we've been building the model using the corn data. So unsurprisingly, when we started using the potato data, the model wasn't as good. So my guess is basically model tweaking, that our model is still, it's been too tweaked to look at just corn. So we need to look at a few more crops so that we can have a model that, you know, navigates among them better. This is all completely speculative. We haven't tried that. That's just my best guess.
Okay, great. Thank you for the interesting work and also discussion. Again, thank you again for the presenting. So now we're going to our second speaker, Dr. Arnold Schumann. Are you there, Dr. Arnold Schumann? Okay, great. Yep, thank you. So I'd like to introduce Dr. Schumann a little bit. Dr. Schumann is a professor of soil, water, and ecosystem sciences. He has dedicated many years in the field of precision agriculture. In the past 4 years, he has published more than 30 journal articles on this subject, including the use of AI in agriculture. His earlier research was focused on ultrasonic mapping of citrus canopy size and employing electromagnetic induction to map water tables. He collaborated with citrus growers in Florida and blueberry growers in Canada to create variable rate sprayers that account for highly variable field condition while saving agrochemicals. In addition, he pioneered the commercial development of variable rate sprayer and fertilizer applicator for citrus that adjust the agrochemical rate according to canopy size. Since 2016, Dr. Schumann experimented with various implementation of deep learning using convolutional artificial neural network to enhance the performance of precision agriculture sensor, especially in machine vision. Thank you, Dr. Schumann, for agreeing to present here. Now, floor is yours.
Thank you, Dana, and good afternoon, everybody. Let's start to share. And just give me a second to get all the menus off my screen here.
Yes, I can see your screen, presentation mode.
Good, thank you. Okay, so I'm going to talk on, I think, I'm trying to make it as interesting as possible, you know, give you examples, but give you a little bit of the differences between two important generative models. So just by way of introduction, I'll show you some of the applications that why is machine vision important in agriculture and why are we bothering to do generative AI to generate better models for machine vision. I mean, why bother? You've got to have a need for it. Then the problems with limited data, training the models. What we've tried so far has been the mainstay methods of data augmentation and synthetic image generation, and then why we want to move to unleashing the potential of generative AI. So kind of the transition, the evolution of the methods, and then conclude. So here's some examples just in citrus alone. You know, machine vision is so important for identifying, diagnosing, counting. Here's blooms in citrus. There's leaf flush, which is food for pests. We can look at defects, off-bloom fruit, which is a green fruit that won't be harvestable at the same time as the main crop. So many things that you can use machine vision for with the right training. Split fruit defects, sunburn fruit defects, pests, more pests, and diseases. And, you know, any crop can benefit from this machine vision. Here's an example from Nova Scotia in Canada, where you can see wild blueberries. These are low bush blueberries. The difference between low bush blueberries and the high bush blueberries that are grown in Florida is that the low bushes are all harvested at the same time. It's not spot picked. So it's very important to get it all at the same ripeness stage, the correct ripeness stage. And so this is a feasibility study we did where we looked at the use of a object detection model to classify the crop into 3 different ripeness stages. The green unripe stage, then the intermediate red stage, and then the final blue harvestable crop. And it's to become a harvesting aid for low bush blueberry growers. These are just the labels. And there's so many. I could go on for a long time, but here's something we've done in Florida with Nathan Boyd, folks at BALM, looking at weeds in strawberry, identifying them, pinpoint accuracy, and then you can selectively spray them with targeted sprays. And then I've also developed, together with my folks here at the CREC, my team and the Precision Ag Lab here, we've developed this app that diagnoses citrus leaves, and I will show it to you in action. It's pretty simple. You just use a smartphone app, take a photo, and send it off for analysis. And if I can get this play here, it should work. Yeah, you'll see 2 smartphones side by side just to save time, and it'll show you 2 examples how you take the photo with your phone's camera. If you've got a problem you see in the field, you pick a leaf, you take a photo of it, and you submit it. And within a second, it gives you an answer. On the left is iron deficiency, on the right is manganese deficiency. You can see very subtle differences, but you can click on more info, it gives you a description, and then even more info. If you click on the link again, it gives you the document in IFAS EDIS to give you a full description of it. And I think I have time to show you the next one here. There's one for pest control starting up on the left. And you'll see why in a minute. Again, there's a problem with that leaf. Snap a photo of it. And so it diagnoses it as leaf miner. All right, so where are we now with citrus and exotic diseases? We're in a bad shape. Exotic diseases are a major threat for not just citrus, but any food crops. And so Florida is a great example of the Huanglongbing or HLB disease has reduced Florida orange production by 90% in just 18 years. And on the right, you can see an HLB affected tree with no fruit on it. You know, that's how bad it gets. So we have to be on our guard. So there's many more. Citrus HLB is not the last one that'll come to Florida. So citrus leprosis is another one, and that's shown in this picture here. Terrible disease also. Now that's not a bacteria, it's a virus vectored by mites, and it's already as close as Mexico. So we have to be on our guard. So adding exotic disease symptoms to our diagnostic app could help with early detection and quarantine action. The problem, however, is that because it's not yet in Florida, we don't have ready access to lots of images for training the model. How do we do that? We need thousands of images, which we don't have yet. So solutions are data augmentation. It's a partial solution. Better yet, synthetic image generation. So synthetic image generation, I just wanted to show you, can be done real simply. It doesn't have to be complicated. One way to do it is to use segmentation and background removal and background replacement. And the idea of data augmentation and synthetic data generation really is that you want to train your model effectively and prevent overfitting. You remember from statistics, degrees of freedom. If you don't have enough degrees of freedom, you're going to overfit. You know, the shortest distance between 2 points is a straight line, 1 degree of freedom. So you want lots of data. And the bigger your model, complex your model is, which is neural networks, the more nodes it has, the more data it craves, right? So it wants thousands, preferably millions of images. So that's just in a nutshell why we even do this. We have to, because otherwise you get overfitting. Even worse, when you do your predictions, you get lack of generalization. And so you'll get wrong answers. So here's just some examples real quick, which I've, you can basically easily do this yourself. You remove the background with segmentation. That's another deep learning model. And then with a little bit of Python script, you can automate the process of randomly superimposing your item of interest, which is your leaves, onto different backgrounds. I just took these randomly from the internet, harvested images from the internet, and off you go. And that helps the model to generalize and to not overfit. So here's the process. Here's my original image on paper. And that's superimposed onto a person playing tennis. I use the model UNet, semantic segmentation. You can also crop it with, you can automate the cropping process with YOLO if you want. Then my ultimate image processing or image classification model that I used for the leaf diagnosis model was EfficientNet B7. So that's where it ultimately ends up. So on the left, if you remove the background, which is that one there with the white, you get very good prediction, but again, it doesn't generalize as well as if you just introduce different backgrounds to it. So I've settled on the replacement map. But here's just a few more examples. That leaf on the left is on some screen, background removal on the right, on a hand, background removal. It's real easy. So that's a partial solution. Now, before I get into this, there's also, of course, all the standard methods of augmentation, and they're pretty much automated. If you do deep learning training on a server with a GPU, you will, on the hypergator if you want, you have the ability to automatically augment your images. In other words, it rotates them at different degrees of rotation. It zooms in and out. It can change the brightness, contrast. It can even blur them. There's so many things it can do, but that is not the same as completely generating new images. But it all helps. It's yet another tool. And that's what they call data or image augmentation. So let's get to unleashing the generative adversarial networks and other generative models. The two and specifically that I want to address is the generative adversarial network. It's the first one. The second one is the latent diffusion model, which I will get to later. So this one really is this simple diagram shows you how it operates, how it gets trained. And the training process really consists of training a discriminator model and a generator model, which compete for attention. They both compete with each other. The generator model tries to generate passable facts that will actually pass a test if you compare them with the real example, right? So it has to keep getting better. It has to keep getting cleverer at generating fake images. Now, the discriminator model, its job is to detect those mistakes that the generator makes, right? So it also keeps getting better. If you keep looping the feedback back to the models, they both learn simultaneously until you get a really good, authentic image, right? It takes a long time. Don't expect GANs to work fast. They can take days or weeks to train. They take a lot of data to. So that's just a basic description there. And you can look at the link at the bottom. But this was kind of the original one. It's the older of the two mainstream types of generative methods. And I wanted to show you these images, again, out of our leaf diagnosis app for citrus, iron deficiency, HLB disease, magnesium, greasy spot disease, scab disease, which is a fungus also, and then citrus canker disease, which is bacterial. So you can see they all look different, but to the untrained eye, not so much, you know. But what I want to show you is, and ask you, what do you think, which row is real and which is fake? The top row or the bottom row? Because they're both different. Here's the answer. Real images are at the bottom. The top ones were generated with a GAN. Granted, it took more than a day to train. Like I said, don't expect massively fast results. But once trained, you can generate tens of thousands of images like this easily. And they're all uniquely different. So the training process, yes, is very slow. Prediction stage is very fast. It takes you, 30 minutes to generate thousands of images. And here's just a quick view of the training stages. This is after 80 iterations. It generally just learns the outline of a leaf. After more stages, more iterations of 240, it starts to learn more details inside the leaf and the shape gets more accurate. And this is finally after about 4,000 iterations. This is the quality you get out of it. It's photo quality. All the reflections and the glossiness of the leaf and everything is in place. However, there are some defects. The kind of defect rate I've been measuring is less than 0.5%. So you probably, if you use this method, it's a good idea to create a Python program that will actually search for defects. If you want to automate it, because you're probably looking at tens of thousands of images. Generate a program that'll look for defects and remove them or flag them, because you do get some that just don't work out. And as I promised, I would show you a lot of interesting examples about all what you can do with this. On the left is a real image of wild radish in wheat. And this was actually from an online database that are, where they offer images for research from University of Sydney. So it's a wheat research site. And I downloaded them. There were thousands of images, and I trained again to generate the images on the right. And you can see it's very authentic, even the shadows and everything. Some of the wild radish leaves are a little bit distorted, not as accurate as the real radish leaves on the left there. But otherwise, the soil color and everything is real good. Here's the lowbush blueberry, like I showed you before. Real image on the left, synthetic on the right. Leaves are real accurate. The fruits are a little bit distorted, but not bad. The color is accurate. And here's so that's the first thing, is exotic diseases. I mentioned you've got to have real reasons to want to do this. Exotic diseases is very important. And I mentioned the example with leprosis. Here's another one: challenges in identifying strawberry diseases. They are very difficult to identify on a field scale, and I'll show you why. Here's Pestalozzi, which is a fairly new arrival in Florida, and at the bottom is leaf blotch, and you can only tell them apart, by expert eye, very expert eye, or by PCR, or by culturing the bacteria, the fungus. So what can we do about difficult cases like that? Well, it's very important to not make mistakes. We need to be 100% certain that the labels that we give the symptoms are correct before we start training models, because incorrect labeling image for training means we're going to have a flawed model and bad predictions. So that's why we have to dig deeper. So we know from scientific background that the best method for positive identification is in the genes, in the DNA. And so we can actually test for the pathogen's presence with their DNA using qPCR, but it's slow and expensive. So that's not the answer. However, it's good to verify. And so because of its cost and its slowness, it's not feasible to test every leaf sample matching 10,000 images that we know we need for training these models. However, we figured it's feasible to thoroughly inspect, photograph, and qPCR test a smaller subset of symptomatic plants and leaves, like 20 to 30, and see if we can use those for generating synthetic images. Because then we know the synthetic images will resemble the real thing, which we know is true because we've measured it with PCR. So we're getting that direct linkage. So like I said in the last line there, to then generate thousands of them, representative of the real deal. And so that's when we moved to, we knew GAN couldn't do that. GAN needs, I've used as little as 600, but you need typically thousands of images. So the GAN was not the answer. I moved to the unstable diffusion network, synthetic image generation, and it works differently. This isn't, just in a nutshell, is how it works. It basically adds noise. So, top line, let me just see what my notes say. Belongs to a different class of generative deep learning models called diffusion. And the reason it's called diffusion is because the underlying mathematics resemble physical processes of diffusion. Forward diffusion process adds noise to a training image, gradually turning it into unrecognizable noise. And this is really well illustrated from this. You can look at the link there, the website that I've given there as a reference. This is a cat, and you keep adding more noise until it's unrecognizable. But at the same time, the model learns how much noise was added, right? So that it starts looking at ways of reversing it. And this is the reversal process, is reverse diffusion, recovers the image, and it does it stepwise. So you have iterative processes, stepwise. It learns to predict the amount of noise and then remove it at each step until it gets back to the original image. Quite amazing. So it works by subtracting the predicted noise from the image successively. During training, the network learns to predict the amount of noise at each step. So that's roughly how it works. The trained model is guided by text prompt conditions to generate the desired image. For example, a photo of a cat. Now, as this website pointed out here, don't be fooled into thinking that it literally occurs like this as illustrated here, because it doesn't happen on a pixel level. It happens on a vector embedded, basically embedded vectors, which is pixels converted into and saved as vectors in the deep learning model. So it's on a different plane. This is just for illustration that I'm using images and noise pixels. It doesn't actually happen that way, but the idea is correct. So we went ahead and tried that. Here's real Pestalozzi on the top, and these are the diffusion model generated images on the bottom, synthetic. You can see it makes some mistakes. A strawberry is a trifoliate leaf. You can see there's actually 4 leaflets there instead of 3. So you get some interesting little defects. But the good news is that it takes just 20 to 30 images to train that, not much, and then you can generate thousands. It takes about an hour to generate thousands, 5,000 here, and here's just a closer view of them. This is leaf scorch, it's a different disease. And I think here I've just got a close-up, real leaf scorch, synthetic leaf scorch at the bottom. And here's a few more examples to finish off. We've got watermelons that we wanted to do a project on, detecting watermelons, and we needed a whole lot more images. There's real on the left, synthetic on the right. That's in plasticulture. It's interesting that when it comes across writing like this is a real image. It tries to also imitate the writing, and it just can't get it right. I haven't seen a single legible, number coming out of the synthetic images. It's always just scribble like that, but it does try. This is a citrus mealybug. It's a pretty bad problem, but even worse is the spherical mealybug. It's more aggressive and damaging. And then we also have this biocontrol. It's the mealybug destroyer larvae, which is basically a ladybug, but the larva looks just like a mealybug. So obviously it's really important to be able to tell. If I had an app that identified them, it would be important for the app to be able to recognize the beneficial biocontrol on the right versus the really damaging spherical mealybug versus the regular citrus mealybug. So these are all real images. On the next page is what diffusion model was able to generate: synthetic citrus mealybug, synthetic spherical, and then the mealybug destroyer beneficial. Here's real and synthetic split fruit, again done with the diffusion model, and you can see you can generate thousands of examples with it at will. Just real fruit with no defects versus synthetic. I can't really tell the difference. It's amazing, reflections and everything it does. I've done this for the same reason as I mentioned before, just lack of data. My challenge was I needed to develop an app to identify this invasive species here. It's very aggressive, the ragweed parthenium. So with the unexpert eye could confuse it with the common ragweed that I'm sure you've all encountered in Florida. This one's fairly rare at the moment, but it does exist. Where it does invade, it's a big problem. So it's important to determine it. But because it's relatively uncommon, I wasn't able to get a lot of images of it. So I generated used the diffusion model again to train and then generate synthetic images. So these are all real. On the top are real. The two top ones are real. Common on the left, parthenium on the right. On the bottom is synthetic common ragweed, and on the bottom is synthetic parthenium ragweed. Nice detail. Even the little hairs show up there on the petiole. And all right, now I've got the slide to change. So to conclude then, some observations about these two main methods here. The GAN models generate less image-to-image diversity than the diffusion models. And I think it's as a result of tending to memorize it. You don't want something to memorize it. You want it to just guess. So the diffusion model is great because it's like a physical diffusion process. It's random number based. So there's much more diversity being introduced. So I'm much prefer that. Like I said, we want to maximize diversity. Diffusion models generate images by starting from random pixels. So every image is more unique from the others. Both the diffusion and GAN models require square images for training. Just keep that in mind. It does not work in rectangular. It has to be exact number of pixels on the height and width, and also generates square synthetic images. Stable diffusion models with conditional text prompts. Well, the great thing about it is it only needs to be fine-tuned with your own custom text prompts and a very small number of images, as little as 20 to 30. You can have more. I've used hundreds. You can apparently use as little as 5, but I wasn't going to go there. I think that's being a bit crazy. So I've used at least 20. Use very highest quality images you can find because everything depends on those few images. And the training is really fast. It only takes about half an hour with a reasonable GPU. So a huge advantage over the GAN also. Very fast. It doesn't take a lot of energy, electrical energy to create a new model. So the GANs, like I said before, use a lot of images, at least 500. They recommend thousands or millions. And it takes days or weeks, even on 8 hypergated GPUs to train. Expert, like I said, recommend at least 50,000. There's the number, or millions, which is a severe limitation, obviously, in our research area and makes the stable diffusion models a much better choice. So you can have some fun with these as well. Here's a pest I discovered here called Ovis aries pests. and they frequent citrus in our area. You can see them, which look a little bit like the mealybugs. And here's some other new life forms we've discovered in our groves. That's a funny crab looking one. This one looks like a citrus frog. So I'd like to acknowledge our team members here in the lab and some funding that we have, and be happy to answer any questions if there's still time.
Great. Thank you very much for interesting presentation. Is there any question from audience? Henry?
Yeah, so ask a quick question. This is fascinating. Very nice presentation, Arnold. Thank you. Looks like we have a lot to talk about. I was just wondering, in terms of applications, so would it make it easier? Because one challenge, I mean, all the generative models have a lot of challenges, right? And one of the big ones for all the models is really, generalization, right? Like you're kind of learning all the different modes of this high dimensional data. So in let's say in the case of classifying diseases, do you think it could be beneficial to first kind of segment the lesions and use the lesions as your kind of, guiding, how can I say, feature to determine that there's a certain disease, or you think that's, looking at the lesions themselves is not necessarily, give you enough information to tell what's going on?
Yeah, I don't think there's a single good answer for that, but that's definitely going to work well for well-defined lesions, like let's say citrus canker, which is a spherical type of lesion with a yellow halo around it. Whereas others, let's say a nutrient deficiency occurs as just yellow veins or something, and so it's not localized. So sometimes it'll work really well, other times not. But yeah, this is why deep learning research, and I really don't like calling it AI research. It's not really intelligent. It's deep learning. That's the correct name. It's very fascinating to me because there's no end to it. You keep opening up more doors and more opportunities and more questions and more answers. And my firm belief is there's always more answers than there's questions. So you will find answers. Just keep looking. But that's a great question. What you say should definitely be tried, but I do believe it won't work in every application. Yeah.
Yeah, so I agree with you. Just if you decide that you'd like to try and segment lesions, let me know, because we're developing a model that should
keep
work
in touch.
really well.
Absolutely. Yeah. Let me know if I can help. Yeah.
Thank you.
Yanis?
Yeah, thank you guys for the presentations. Both of them are excellent. Arnold, we are using GANs also to generate synthetic data. So then we found out that like the relationship between the real data and the synthetic data has to be at least you use the same amount of synthetic and real. If you use more synthetic and then you try to evaluate your models, we found more errors. Is this what you see also? I mean, the question actually is like, can you just go and create synthetic data and use only the synthetic data later to train your models and use your models? Or you try to have a balance between real and synthetic?
Yeah, I know exactly what you mean. And it's something I've tried out multiple times. So, but in the process of that discovery, I've also, like I said before, there's no single answer to that. And I can just give you some examples. If your training data for your synthetic model was super good, I mean, super high quality, you're probably going to find that you can stretch it further and use more than double, like you said. So that's one factor. And even your let's say it's just a simple classification model. Starting point will matter too. If you start off with only 100 and you double that with synthetic images to 200, you'll get different answers compared to when you start off with 600 real images and double that with 600 more synthetic images to 1,200. So the example I can absolutely in recent memory refer to is that I use 600 starting images for 6 different classes and trained it just with those real images. Then I generated increasing amounts of synthetic images all the way up to more than 10,000. And I found that at 10,000, there was an inflection point that plateaued. So So in other words, what you found is the true answer in your situation, but it won't necessarily be the same answer in a different situation. Likewise, the result I got won't be applicable to all situations. It'll be, you have to just try it out whatever you do, just try it. And so one thing I can say also is that my validation set, and of course I only look at the validation accuracy, I don't care about the training accuracy when you add more synthetic data. So my validation accuracy, like I said, increased up to 10,000, but I gave it a very tough job to do. My training and my real and synthetic data was all without the backgrounds, like I showed you those leaves. But my validation set was a completely different set, which it should be, taken on a different year even, and with the background still in place. So it was a tough job for it to do because it hadn't ever been trained with those backgrounds. And again, I think that influenced the benefit I got from synthetic data. So I shake my head because there's no real, I can't give you a definitive answer, but I'll give you all that background just so you understand why.
Yeah, thank you. And I totally agree. And from our experience, what we see when you try to use the model for even like in different locations, and sometimes there are different varieties, crops somehow. I mean, this is where the problem starts. So, but you're right, it's not easy to answer it. Just I want to bring it up because it's something that we need to consider.
Sure.
Okay, great. We're going to have one more question, Choi.
Oh, great, thank you. Excellent talk. So it seems that the image or machine can really learn the detail of the problem. So for instance, those even the hair on the pedicels, their machine were able to capture this subtle changes. So the question is, if we can provide high resolution images, is the machine were able to capture those very small changes for instance, even subcellular or even cellular level, the detail that is it possible to reach that level?
Yeah, all the tools are there. I think you might have to look at concatenating a bunch of models. Like for example, you get other generative models that increase resolution. So the problem with and I call the ones I've used the basic models, both the GAN, which is style GAN I was using, and then the diffusion model, they're both limited in the resolution that they can manage for training and for prediction. So typically, the one I've only worked up to 1024 by 1024, which still isn't a high resolution. The stable diffusion prefers 512 by 512, or depending on which version you use, it can go up to 768. Of course, that takes tremendous amount more GPU memory, footprint on the GPU, and also CPU. It multiplies up obviously exponentially. So there's those limitations. But if you look at, there's so many ways like tiling that we heard in Henry's talk previously. Using patches and then reassembling patches later on. You can explore those type of techniques. Moving kernels, windows, whatever you want to call them. There's always an answer. So don't give up if you've got a real challenging question, and let's work together. Let me know if maybe I can help in any way. I think answers can be found for all those issues, and there will be.
Great, thank you.
Great. Thank you again for both of our speakers. It was a really excellent talk. I'd like to conclude our seminar here today. Next seminar will be December 8th. I'm going to follow up with you with a flyer with more detail. Thank you again. I'll see you next time.
Thank you.
Everyone.